but I will do it right now.
Okay, I just clicked now. Okay, I won't be reading the screen. I'm not able to read the screen, but I can answer questions, discuss matters. So why don't we start with who's Sally?
Um, so I will give update. So first of all, just a weekly update about EBW. The first screening, we have around 937 patients left. So we're finally under 1,000 points. So hopefully we'll keep going in this pace so we can try to get it done at probably before the summer. That's our goal.
Okay.
And then project update. So
Wait.
Eugene,
The EDW is the undergrad one, right?
yes, the first screening.
What about the MD portion? How are we doing on that?
So second screening, the new doctors here, they just learned how to do it yesterday, and they tried on their own, one patient. So like I think today they will try it again and get to the same point because I think there's so many things that they need to like discuss and talk about to make it consistent throughout. So
Mm,
yeah, Dr. Wongi, he gave them orientation yesterday.
okay.
And then I know Arnie is working on it, and Monica's probably busy with her wedding, and then Dr. Wongi is working on it. So, yes,
The
I'm not
second
too sure
screen.
about the status. Yes.
Uh, Liam, can you give me update on where we are, how far we have to go?
On the second screening?
Yeah.
Yes. So, um, we are so far we are working on a list of 250 or so patients, and I believe we are about we've finished about 50 so far. These are done from the previous doctors, like when Dr. Seungmi was here or when Chae he was here. And Arnie's working on it, and then we have the three new doctors who started today. So we should be making some progress, but it takes some time. Initially, it takes like about an hour on average for one patient. And but in our experience, like last year when Dr. Jihan was here, she was able to do about one every like 20 minutes as she got used to the process. So it'll just take some time for the new doctors to get used to the system, but they started working on it yesterday, and they were able to finish one patient each. And I think they'll be able to pick up the pace as we go through the week. Yeah, so we have so far about 1,000 patients left for the first screening, which is being done by the students, and that means we'll have about 900 patients remaining for the EDW for the second screening. And yesterday we discussed with Dr. Wangi about collecting all the CT scan dates for the radiomics so that we can request the DICOM files after we do the second screening. And the
Mm,
same is true for the pathology accession numbers, so we can request the pathology slides as well at the end of the process.
right. Make it a simultaneous process. Yeah, I agree.
Mhm.
Do
So
you
we're
have
working on that.
any update on Dr. Isul's project with pathology?
We have a meeting. Well, we're trying to schedule a meeting. So Dr. Isol and Dr. Sinha, they're still working on updating that list. And Dr. Batosh says he should be able to pull the slides for us in about a week once we send over the finalized list.
So I thought we were done with the list on that Wednesday. What happened?
We were done with we sent him the list, but he still wanted us to like differentiate if these were like FNA samples or these were surgical blocks,
Oh,
and also to pinpoint like which block the stain like the H&E samples were collected from. Basically, our understanding is he just wanted us to pinpoint the slide so that it would minimize his workflow and make it more efficient.
so that's doable on our end. We can search for that.
Yes, so we've been working on it. Last I spoke with Dr. Isel was last week. And so we've been going through all of the path reports one by one and identifying the block numbers if relevant. So some of them don't have multiple blocks. So if it's just one block, we left it alone. But then if there are multiple blocks, we chose the correct block that was used for the staining and slide.
Hmm.
Yeah, and Dr. Batash requested to have like a short meeting sometime this week if possible to discuss, and we're still in the process of scheduling that. But I think everything is in motion. Dr. Isel said she'll be able to send the list um, with like the updated information sometime today or tomorrow. And Dr. Batash said he should be able to pull the slides in about a week once he receives them.
Okay.
Yeah, there were several email communications that went back and forth yesterday.
Okay. If we can have that meeting, well, I think Dr. Isol has to be present because she's been looking at it the most. So,
Mhm.
and depending on her availability, I'll either, attend or not attend, depending on my availability. But I think they both need to be present.
Mhm.
Okay. Okay. Um, okay. Let's go back to Sally.
Yeah. So yeah, so that's it for EDW. And then this is for VC projects. So I'm still in contact with Eugene, and thankfully we're in good communication about what's going on. So they submitted the revision manuscript to Helion, and he's trying to submit it through preprint as well, just in case that, it might take a while for Helion to process the revision. So he said he will do it by the end of this week. So that's it for his project. Grace Lee, it's the same. I don't think she has time right now, but she says she will try to work on it near future. And then Emma's project, so submission is still in progress. So last update that I heard was that they finished up with making more tables and figures, and now they're like ready to submit. So Eileen was actually working on submitting submission process last like two days ago, or yesterday when she was here, or Friday when she was here. So I think that's still going on. And then lastly, today I wanted to show you the revision comments, but you're not able to read, right? Should I just read it off, or would it be better if we do it later?
Is it is it a long comment or is there a question or what do you think? I
So
can't.
yeah, so I can just quickly share. So
Yeah.
their comments were basically, um, they said our prediction tools evaluating the study are not considered solid evidence for variant classification. And they said they're not sufficient description of how ChatGPT yields like useful prediction for these variants. So Dr. Yuni and I, we weren't sure what to do about this comment.
But there's no suggestion. It's just comment, right?
Yes, it's just comments on how like they don't think there is sufficient description of how like ChatGPT can be a useful prediction about classification, and then also like um they're not considered as like solid evidence for variant classification, the tools that we used. Should we just explain
So
in more details?
so what this reviewer's comment is that a major revision or what, where are we?
Yeah, major revision.
Major revision. So if there's no question or suggestion, that we can't answer that. It's just their thought.
And then they said that only like 160 missense variants is too small of a sample
Uh-huh.
for this type of study. So I think that's suggesting that we should add more samples. And then they also commented on how like the title of the paper is misleading. Because we said like evaluation of in silico tools and ChatGPT to determine the impact of missense variants and immune-related gene associated with immunotherapy outcome for solid tumors. And they thought, they said we only looked at like non-small cell lung cancer tumor variants. So they thought it was misleading. So that's maybe we could change that, but yeah, mainly I think they wanted more samples. And then yeah,
So
and mhm.
so immunotherapy related genes were selected and those criteria were described in the method.
Mm-hmm.
Now, tell me what happened to the non-small cell lung cancer. We only looked at lung patients.
No, we did any genes that were related to.
Right, but what is the comment about? I'm trying to understand.
Oh, the comment says like all the variants that we looked at are from non-small cell lung cancer cases
Cases.
or like tumor variants, like only non-small cell lung cancer tumor variants are examined.
Only lung cancer. So tumor variants found in lung cancer, is that what they're I'm trying to understand. Because we didn't write this paper based on lung cancer cohort or anything.
Mm-mm, we did not.
Okay, for that comment, we can say the genes selected for this paper are based on the commonly the most commonly reported genes and non-small cell lung cancer relate patients data. I don't. Well, the lung cancer was not the selection criteria, and
Mm-mm.
it happens to be that uh, for the selected genes, there are more literature supporting the the role of the gene mutations in different in affecting differential immunotherapy related outcomes, something like that.
Mm-hmm.
It's not like we selected only lung related genes. Just say that in a more modest way.
Okay.
And then title, why is title misleading?
Because we
I don't.
said solid tumors, and I think that's where they got confused because, um, yeah, because we said like we looked at the impact of like missense variants of immune-related genes associated with immunotherapy outcomes for like solid tumors. And I think that's why for them in their perspective, they
Oh,
thought, yeah, all the variants were related to non-small cell lung cancer. So why do we
so
say solid
now if you go back to introduction or discussion, maybe we cited only papers that are
non?
like lung cancer based. Maybe that's where the misconception is arising.
Mm,
So check that. And renal cell, um, melanoma, you can add more citations that so the genes that we selected do play a role in other solid tumors. So maybe add a couple more solid tumor evidences that because genes are not lung cancer specific. They are present in all immunogenic tumors that have immunotherapy FDA approved.
mhm,
So look for that and find some related studies and add those citations and say in the comments, those we've all, those are also genes related to differential immunotherapy outcomes in other solid tumors such as melanoma and renal cell. Um, I mean, there are others like MSI high, colorectal cancer, cervical cancer, head and neck
mm,
cancer, liver cancer. Uh, these are all tumors that immunotherapies have been approved for. So you
mhm,
could say that we've added new supporting um, sentences in either introduction or preferably in discussion and say in this, discussion section, this is what we added and just copy paste what you added in the
mhm,
discussion. So that
right.
let's
So
go
what?
back to the title. Can you read the title again?
Yes. Evaluation of in silico tools and ChatGPT to determine the impact of missense variants of immune-related genes associated with immunotherapy outcomes for solid tumors.
Okay, so it is solid for cancer for sure. And we should describe why these genes have, what are the literature that supports the fact that these genes have shown differential outcomes in immunotherapy. Like STK11 is not only important for lung, it can be important for melanoma, PTEN can be important, PTEN loss can be important for melanoma immunotherapy, things like that.
Okay.
So, so because it's not lung cancer specific, the title is not misleading. But don't say it's not misleading. Just say, we still we do believe that these genes are not lung cancer specific, but that are also pertinent to other solid tumors, and we've added those evidence to say it politely.
Okay. Okay. Yeah, we can definitely
And then
do that.
ChatGPT. uh we can defend ourselves by saying we understand that some um people may resort to ChatGPT as a new tool to ask this question: Are these specific variants functional or not? For that real world, practical concern, we tested ChatGPT's I guess role or uh, performance and assessed it with the same metrics, which are sensitivity, specificity, all that same metrics. So same metrics were used. It's not like we didn't use scientific rules to validate it. So,
Mm,
it but ChatGPT was not chosen for its scientific rigor, but rather it's chosen given a concern uh, whether this can or should be used to answer these type of questions.
Mm-hmm.
more like real world setting. So
Yeah.
so we can
Okay.
kind of add that comment.
So a comment about how like 160 missense variants are too small. Should we add more, or how should we go about that comment?
160?
60, yeah.
Okay. So I think if we get more, then I think we have to add more patients, and I think we just have to justify, based on the selection criteria, this was what was chosen. To say it how we came, it's not like we want only 160. It's described based on our selection criteria of genes. Even if you want to get more, there may not be more functional variants that
Mhm.
we found. Something like,
Yeah, we tried to originally we tried to get more variants,
yeah,
but these were the only ones.
yeah, we can say we aimed for higher number, but given this, scientifically rigor, not rigorous, but given this clear guide um selection criteria, we were only able to get some, just describe why we came up with that number, and that's it. And we don't, unless they like, please add more, gene, we can't do it. I don't think we have bandwidth to do that now.
Mm, okay.
Okay.
I think those are like major things that we had questions about, but yeah, I
Reviewer
think
2
that's
was okay. Reviewer 2 was okay.
oh, those were so I combined them in one question or few
Oh, okay.
questions. Yeah. But there are like other minor comments,
Okay,
like yeah, so I can take care of that.
sure. And this is what journal?
It's we did it in translational cancer research.
Okay, good. Um, yeah, I mean, it's good that we actually got revision for it, so
Mm-hmm. Yes, it's going.
yeah.
Yeah, so that is it for BC projects. Um, yeah, and then RedCap, we're still updating. So Dr. Liam and I are working on that together, to possibly, yeah, we are trying to, yeah, once we're done, like we'll bring it to you so you can take a look at it. And then dream patients, we're constantly trying to reach out to them to get consents from them. So that's still in progress. And then there's one more. Oh, yeah, that's it. And then Dart trials and last trial, I think Dr. Liam will update.
Mm, mm. Who's writing? Oh yeah, I know Tarek is writing case report for Nabor, which is our first adrenocortical patient. Our second adrenocortical patient is still waiting, I think waiting for lung. I forgot her name. Liam, do you know her name?
Not off the top of my head. I'll have to look it up.
Yeah. So recently there's been a couple patients who got lung transplant. So update your list, get the list from Brooke, and keep annotating their pathology and all that. So here's what I want to do. There are patients. There's Von Imes. I think I saw that patient with Dr. Sunga, and um, Michael Goldstone as well. Some of the patients that we thought was cleared of cancer after transplant, they are found to have cancer. So I'm doing adjuvant chemo for them. But anyway, they're not technically cohort A or C. However, we should so they shouldn't be added to A or C because before transplant we didn't have any even history of cancer or evidence of active cancer. So that they're not A or C, but I still get to see them. But we can still have them consent for DREAM, but not and just leave them as an ineligible patient, and still track their outcome because we also want to find out what happens to these patients. That's the only reason that we want to track them. It's a tricky case. And it's very unfortunate to see Mr. Im's case, where right after transplant, patient has bone mets, adrenal gland mets. They're very frustrated. So, but I wasn't able to see the patient because there never was an evidence of cancer before the transplant. Anyway, so what I'm saying is let's keep collecting those patients as well. So Sally and Liam follow up with Dr. Tarek to write the case reports together for cohort A, cohort C, and cohort B. There's only one patient, but start writing it now. It's better to do it now. And we want to write even a case report or case series if the second patient gets lung transplant. And yesterday I went to see Mr. Nabor with Dr. Sinha, and we heard a beautiful story. And you can hear the story from Dr. Sinha today. But I just wanted to share one aspect that the patient was persistently asking his medical oncologist in New York City for a double lung transplant. So it wasn't just Albert Curie. We had another Albert Curie in New York, and Dr. Foho was the medical oncologist treating adrenocortical carcinoma. And he, just like myself, he got convinced that double lung transplant is the next step. And he looked for any surgeon who would operate on that Mr. Nabor and found our program and referred him to us. But if it wasn't for Albert Curie, was very persistent also asking me for a double lung transplant, we, you know, we would not have, made this dream program. So this, amazing interaction gave me goosebumps yesterday listening to his story. But I wanted to share it with all you guys because Dream Project is one of our priorities in our lab. And being a pioneer, we want to keep publishing, presenting these data, and we believe there are many cohort A or cohort C patients out there who just don't know that transplant is their option. And it's so unfortunate and sad to think about those cases. So the more we publish, present, and the more we study the biological rationale behind these special type of tumors, the better these patients will be served. And the medical community will open up to these what seemingly is radical, but rather innovative um, uh, treatment method. Okay, so that's my comment for all of you guys. Okay, any other updates for Dream?
I think that's it, right? Dr. Liam? Yeah.
So
Yes.
again,
Yeah.
touch base with Tarek like weekly basis. Make sure you know where he is in writing up these chord ABC. And write the paper together. You're not there to just edit. Write it together with him. You guys are part of the Dream Project together. Brooke wants to be part of the paper. Let Brooke write some part of it. If she wants to be an author, she has to work for it. So just be a team leader for writing any publications for Dream Project. We want to keep publishing it. So the other thing about Dream Project is we want to write a proposal to Genomic Insight. They changed their name to, what was it? Do you remember? Was it some cross? Oncocross? What was it? Inocross?
Something like that.
Inocross. Anyone remember? Dr. Sunga or anyone? No?
Inokrad.
Inocross or crop?
Inokra.
Anyway, um, Dr. Liam, why don't you take the lead on writing up a one-page proposal? So, to them about the Dream Project. So what we want to do is exactly what Charlie Swanton did with his autopsy paper. Like, you can read his phylogenetic tree papers where using autopsy he drew many phylogenetic trees. So our question is, in patient in cohort A, we want to understand their clonal diversity, phylogenetic um, uh, analysis. Are they all same clone? Are they all different clones? How are they evolving as they seem to spread through airway? So the title would be Clonal Architecture of um, Bilateral Lung Limited Malignancy um, who bilateral lung limited lung adenocarcinoma uh, who undergone um, bilateral lung transplant under Dream program or something. And then the idea is get representative samples from all different lesions within bilateral lungs and analyze it through whole genome sequencing to explore the genomic architecture of those lesions. And the method will be same as what Charlie Swanton did, doing those genomic analysis, and they have informatics people who are able to do that. So we'll start the pilot project with maybe 6 patients that we have. We can include the pneumonectomy patients too. And yeah, and for the lesions, you can look at the CT scan of each patient before transplant and get the number of the different lesions per patient and multiply by patient number. Yeah, so what we're asking for is they'll be able, with a collaboration where they will be able to sequence and do the analysis together with us as a pilot, with the intent to apply for larger grants, like R01 together, for the upcoming enrolling patients. But what we have so far, we can create data to make a preliminary data or pilot project. So we can write that proposal and we can share that. Once we write that, we can share that with Dr. Barat, because I know he's doing something similar, but I know he's not doing whole genome. So doing whole genome will give us an edge. What whole exome cannot do, we can do that with whole genome. So Dr. Liam, work on that and let me know how it goes, okay?
Okay.
Okay. When are you going to the European Lung Congress?
That'll be on the 20th.
Of this month?
Yes.
Wow, it's coming soon. Okay. Oh, pacemaker manuscript, where are we with that?
So I still have it. My apologies on the delay. I'm still like, there's a lot of edits that I needed to put in to the current draft, so I'm working on it.
So what's needed? Is it more technical or do you have to read and write?
It's more reading and writing. It's the formatting piece. So what we have right now, I just want the paper to be more scientific, like the flow and the style and the formatting. So that's what I'm working on.
So that part you have to do it, right? I mean, people who's
Yeah,
not
I
familiar,
think
it's
so.
hard to help, right?
Yeah, it's definitely
Any
gonna be
uh-huh.
yeah, it's definitely gonna be hard for the students to help with this part of the process. Maybe Sally, since she's read enough papers during her MPH, but yeah, it's something that will require
Uh-huh.
our input.
Is Sally part of the co-author?
Yeah, I
She
already
is.
went through it, and that's why I think final step, I think we wanted Dr. Liam to look at it. I already looked at it with a
Oh,
couple of weeks ago. Yeah.
okay. Yeah, I don't know. I mean, you have to, I guess you have to just yeah, revise it and submit it. I
Mhm.
feel like it's been too long, so yeah, let's get it out.
Agreed. Will do.
Yeah, and if you need technical help, you can still work with any students or other doctors in the lab if they want to. Okay.
Of course.
Okay.
Dr. Liam, do we know which journal were submitting it to?
Yeah, I think it was the first one was like JCCN, something like that.
Okay.
It's in our manuscript.
Okay. And we have to format it in that way too.
Mhm.
And this was a survey for healthcare professionals.
Yes.
So does this need any IRB exempt or some? I'm just thinking out loud. It's not patient.
Yeah, you're right. I always assumed our retrospective study covers everything, but it might be outside of the scope of the IRB.
Yeah, maybe ask some questions to IRB personnel that, we did, a small survey just among doctors, nurses. Just ask them what we need for those studies are relevant or not relevant for IRB, or I don't know. I haven't done any study like this. So.
Okay, yes, I'll check with the IRB.
Okay. Okay. Yeah, and then for all of you, Dr. Arnie and Dr. Eugene, Dr. Jian as well, they're the team working on MRD as well as the hyperprogression in our Northwestern patients. So both of those papers are now finally accepted. So yeah, it took them a while, and recently Dr. Min Jung with her meta-analysis for hyperprogression. So after 35 rejections. So guys, uh, it's a daunting process. Requires persistence. But you'll get there if you don't give up and take ownership. No one will do it for you. You have to be persistent. So congratulations. I know they're not here today, but hopefully we'll just celebrate them together in spirit. Okay. Any updates? Well, we'll just hear from the team what you guys learned so far. Dr. Sunga, I know you've learned mostly in clinic, but any comments?
I'll talk about later.
Okay. I know you guys just arrived. Dr. Yu Jung, any comments?
We just started to learn how EDW works and then about the second screening.
Mm.
Yeah, I think it's very thankful to learn about the research. Yeah.
Thank you, Dr. Chongho. Any comment?
Yeah, I'm planning to do some research for double lung transplant. I mean study.
Mm. Oh, you're interested in double lung transplant?
Yeah.
Good.
It sounds new.
Oh, yes.
And also, I was curious about adrenocortical tumor.
Yes.
Yes. So I'm planning to study about them.
Yes. Why don't you, if you like it, why don't you reach out to Tarek, meet with him, and write the paper together?
Um, okay, yes, sir.
Yeah. Liam can tell you how to meet Tarek. So the first 2 weeks for all of you, try to find out what kind of projects are ongoing in our lab by listening to lab meetings, listening to Dr. Liam and Sally, like what things we are doing here, and try to understand why we're doing what we're doing and what are the methodologies, platforms we use to answer those questions. If you're interested in statistics, we can let you be involved in statistical, more bioinformatics projects. There are a lot of clinical projects too, so you'll learn how valuable it is to get data well annotated so that we can trust our results of each analysis. Okay, good. Dr. Soin?
I think it's same for me. I just learned how to do the EDW, and I think going through the charts of the pa um patients of the lung cancer was it gave me a lot of thoughts and how they have to try different chemos or immunotherapies and still sometimes it doesn't work and patients give up their lives. And also listening to the project, I think I'm interested in the like whole genome sequencing of the double lung adenocarcinoma. So hopefully I'll get some chance to learn how to do those things.
Okay. Let's do this. We did this in our lab, but for your and everybody else's own learning, Charlie, or I call him Charlie, but he's Charles Swanton, is a UK medical oncologist and physician scientist like myself. He's also a lung cancer doc, and he wrote many Nature papers in the same issue, which was amazing. He's sort of my role model. And Charlie has um, the clonal analysis paper in Nature or Science. I'm blinking, but look for that paper. I think Liam might know that because we did journal club on it. Why don't you study that paper and maybe 5-minute brief presentation on that paper for next journal club, and I can give you some ideas about our proposal. You can write the proposal together with Dr. Liam, so that you know what we're trying to do. And in the end, if you're interested, the whole genome is interesting, but in the end, what we're trying to do is how do we better understand what's clonal, what's subclonal, what's the genomic diversity? In other words, what is genomic heterogeneity? So I want you to read the review paper from that I wrote with Stas, our medical student. and myself, and that will give you an idea of what why we're interested in this question, heterogeneity. So read that paper, but you'll do journal club 5-minute presentation on Swanton's paper, and let's work on the proposal if you like that. Yes.
Okay.
Any questions? Okay. Liam, any other updates?
Yes. Did you go to Dr. Rachel?
Dr. Rachel?
He did.
Oh, okay. Okay. Give me one moment. Let me just pull up my. So there's a lot of like follow-ups from our outside collaborators. I can share the screen, but I know you can't read your screen right now, so
Yeah, just ask me the question. And if it's something that you can just talk to me in person, that's fine too. But if it's something that I can have a teaching moment, or it's fine to go over it now.
okay. So most of these are a lot of these are related to contracting. Yeah, actually, so yeah, these are all related to like outside company, like
Contracts?
yeah, contracts, documents, and questions regarding contracts. So I think, I mean, but there's a lot, but probably better to go over them in person when you come in. We
Okay.
do have a couple of meetings later today that are going to be important. So we have the last trial correlatives meeting with our Northwestern Path Core manager to discuss our lab protocol and lab manual for the last trial. So
Okay.
this will be the first meeting we have with them to just start the discussions on that regard. That's going to be today at 2 o'clock, I believe. And then at 1:15, we have the meeting with the Tempest people. We're trying to kind of recalibrate the scope of our, our collaboration, the project we have with Tempest. They've been kind of pushing back, saying that if we want to change it, we need to start up basically a new proposal. And we sent them in response saying, we don't think it's a completely different project. We just want to try to work within our current scope, but like, can we be flexible? But I think we'll be able to discuss that in more depth today at the meeting.
Okay.
And then let's see. And then we have the regular last trial like follow-up meetings later on as well, but there won't be much updates there. And Hugh sent us a couple of questions that we can go over later together as well.
Okay. But you got the Kisobio IB, right? So you.
Yes, that one I received and circulated to all the relevant people.
So can you follow up with the product provider? What's keeping her at this point?
Sure. So she said she's still working on a couple of things. We got the IB, so I think she's working on it. I'll follow up with her.
Okay.
And she'll also be present at the meeting today with the PathCore lab people.
Oh, okay. Okay. So last trial. So I want the new members know about what last trial is. Sally, can you tell me the acronym of what the acronym is? The original title. Or Liam.
Yes, so it's tumor infiltrating lymphocyte directed anti-CTLA-4 and anti-PD-1 therapy for all solid tumors unexposed to immunotherapy.
Yeah. So this is our new trial. And once we activate it, we want to be able to do the same thing, you know, do the trial in progress and let people know there's such trial. So Sally and Liam, let's work on finalizing the sister sites together and reach out to them. Let's have have that as a priority to be done today.
Chae, Stas is also here.
Hello.
Hey, good morning.
Good morning.
I'm still on my way to my office.
Got it.
I'm one minute away, but you can start.
Yeah, I mean, for me, the only update is really just I made the changes to the paper that we talked about last week, and I focused it more on how low TMB is the driver versus this is only present in low TMB folks. So that's that's kind of the biggest change.
Right. Right. And you change it to like according to or by TMB because our
Correct.
analysis is not confined to just low TMB, but we looked at both.
Correct.
So okay. So what paper are you submitting it to?
TLCR, Transitional Lung Cancer Research.
Oh yeah.
Translational Lung Cancer Research.
Translation. Okay.
Yeah.
Okay. So
And then
almost,
I'll send it to, yeah, go ahead, sorry.
almost ready or?
It's ready to submit. I just need to kind of make an account, go through the portal, check all the
Oh, okay.
boxes,
Right.
maybe pick some reviewers and editors and stuff.
Okay. Okay, if they have a mandatory review panel, then let me know. I can
Yeah.
find some lung cancer doctors.
So I can start that process today. I'll send the draft to Hasong one more time, have her look at it in case there's anything that stands out to her, and then I'll start the submission process.
Yeah, good. So remember in last meeting with Genomic Insight team, I told you about the dream project.
Yeah.
So we have Liam and Colin here work on it, but feel free to be involved as much as you want because it's about, doing sequencing to, look at all these different intertumoral lesions in mucinous, mainly or lipidic lung cancers
Yeah.
that undergone bilateral lung transplant. So the analysis we're going to do is really whole genome based phylogenetic tree. So yeah, you could be part of the project if you want.
Yeah, I'm
You would,
definitely interested in being involved. I think it's pretty rare to have a project where you have a large number of intertumor lesions. So this will be really
right,
interesting. Um, I'll just look into building
exactly.
phylogenetic trees and how heterogeneity fits into it and kind of the computational side of things. If there's somewhere
Right,
where the data
right.
is stored or if there's something available that I can start working with, that would be great. If not, I'll just wait.
Yeah, so if you go back to the Nature Charlie Swanson paper, you will see they did just like, or the MSK paper, just the
Yeah,
phylogenetic tree, the clonal and subclonal, and they just looked at the percentage. There's kind of primitive way, but there may be more sophisticated way of doing that. So yeah, look into that bioinformatic aspect. How do you um deconvolute
yeah,
that phylogenetic tree into like one heterogeneity index, right?
right.
Yeah. And this and yeah, Stas and Soyin, you can read. I'm trying to find that paper for you guys. That paper, and I talked to Charlie at last year's AACR, and he said he had exactly same patient like us, where mucinous just spread through airway, Stas. And he did look at phylogenetic tree for that patient, and he said he added that in supplemental uh, file. I wasn't able to find it, or I didn't look into it yet, but that's something we could kind of look at and get some ideas from.
The question I have regarding that process, so in those patients, they're not finding any Mets in the lymph nodes, correct? They're finding all of these lesions, but no lymphovascular spread.
Correct. We
Okay.
had
I'll do some reading about it, but that's really interesting.
yeah. Liam, did we ever have hilar lymph node positive patient? Maybe we did with one patient. Can you look?
For a dream patient?
Yeah, cohort A. Were they all lymph node negative? Maybe Sally, you can look into that.
I'll have to check. I don't.
Yeah, maybe you can look into it. Let me know later.
Because I think kind
Okay.
of the most simple
Mhm.
approach that folks have taken is they'll look at the presence or absence of certain driver mutations, and then based on
Mhm.
that, they'll build a tree. But
Mm.
that's I think that's a good method, simple method. It makes sense, but there's probably a lot of subclones that are missed by it.
Right. So I guess the more place you biopsy, the more tumors, intertumoral lesions you include, you will see more trees.
Yeah.
Yeah, it's inevitable that way. So I found some papers, so I will share with all of you guys. Uh, So there are some papers here. I'll put in the chat room. So first paper started with this NEJM paper. tracking the evolution of non-small cell lung cancer. Their autopsy program is called Tracer RX. And then in Nature, he got the evolution of lung cancer and impact of subclonal selection in Tracer RX. This is a seminal paper. For us, we're not looking at therapy selection where everyone will get cured by doing transplant. So it's a little different in that regard. So these two are the seminal papers. And this is not a paper, but this is a nice concept paper written in Cancer Discovery. It's a journal that I like. So you can read these papers. Okay. Anything else, Stas?
No, they used PyClone in that first one actually, which is interesting. But no, yeah, nothing else.
So let's just go through your projects one by one. So that paper you'll submit, that's the
Yep.
TCGA. And I know with Hesong Tempest heterogeneity, you're working on it, right?
Yeah, about a week ago, I replied to an email about setting a time to meet with them to talk about our findings, to hopefully get them to broaden that patient cohort up to 500. And I haven't gotten a reply.
Okay.
So I don't know if there's higher priority stuff that they're working on or what, but I guess I can bug them and remind them.
Okay. Where are we with the garden getting the raw data? Any updates there? Liam or staff, did you hear anything?
Nothing on my end. I mean, the only thing that I've had access to is the spreadsheet that you can download that has the 3 or 4 or 5 at most detected variants. That's about it. That's all I've seen, unless there's something else.
Liam, any updates there?
So we received an email, I think last week or so, saying that the contract just got signed.
So can you follow up? How do we actually get the data now? Or add Shelly Marino? I don't okay. What are the other ones? Tempus clonality, our own patient. What do we decide on that, Stas?
That's a different data set than the one I've been working with from Tempest. Or you mean more generally, what are we going to use?
Oh yeah, so the Hesong's Tempest project was looking at Tempest data as a company, but
versus what we have through the
yeah,
S3
versus
bucket.
our own Northwestern patient data.
Right. So there's a lot of patients there. I don't have a number. I can look back into my notes and see how many of the patients that we have clinical data for or have curated clinical data for. Also have whole exome sequencing and have RNA sequencing. But really, it all comes down to what's been curated clinically, and then I can check if the data's there. And then based on that, we can go ahead. The nice thing is that we can do PyClone on that data. We can do math. We could do also kind of we're free to try whatever we want. So
Yeah,
it's up to
yeah.
us.
Are you talking about Tempus data? Which Tempus?
Correct. So yeah, so Tempus gave us access to an S3 bucket that has all their sequencing. Most patients have DNA and copy number variation. Some patients have RNA, and then even a smaller subset also, I think, have liquid biopsy.
Okay.
But the exact numbers of the subsets, I don't have that, but I can get that information once I have a list of patients that we're interested in, or have clinical curation for.
So this is the company data you're talking about?
My understanding is it's our data from Northwestern patients. Maybe there's patients in that data set that are from other physicians other than yourself, but I think it's all Northwestern.
So these are well,
there might be overlap between the two. I don't know.
So the abstract that you submitted,
Yeah.
that's our Northwestern data.
No, that's the data set that Tempus gave us access to through their workspace. Through Lens.
Through Lens. But that's but those survival outcome and okay, so the treatments and everything was annotated by them.
Correct.
Okay, so that's the company data. And even with starting with 500, we ended up just like 70 or something, like smaller number.
We had less than 500. So we had 500 total between squamous and adeno. And I think adeno had less than 200, and then squamous had a little more than 300, I believe.
I see. So that's the Tempus data. Okay.
And that's the one that Hesong wanted to expand, or at least the adeno cohort wanted to expand that cohort to 500.
Right. So yes, so we do want, forget the squamous now. We just want to look at adeno 500, see what we see in our analysis true with the larger sample size. So we're going to be persistent asking that question. Okay. That being said, I know just our Northwestern. So that 500 is not Northwestern. Northwestern is just a really small part of it, maybe. But what we want is just our Northwestern data. Tempus, probably they're all just my patient because I'm the only lung provider that probably orders Tempus. So I don't know, maybe 50 patients, or have you looked at that yet or not yet?
No, I haven't looked at that yet.
So Liam, can you work on that to get the Tempus data in the, let's say we have radiomics cohort, right? So among them, who has the Tempus data? I think we looked at it.
We did. I think with that cohort, what I did was I looked at who had of that cohort who had data in Tempus, and I think it was out of the 200, I think it was 50 to 60 patients. And we didn't see any statistically significant associations. And then we tried to define clones versus subclones and see if there's any relationship there. And again, the sample sizes got so small that there was nothing statistically significant.
I see. We at least looked at that. Okay. Now we're gonna do something similar with the garden. Once we get the raw data, it will be the more than 300 genes because blood TMB can be calculated from it. So using that, the clonality in blood, can that have any impact on survival, therapy
Yeah.
outcome, things like that. Okay, so we'll still try to do that. Okay. Stas, are you planning to go to any future meetings? ACR, ASCO, or
Well, it all depends where I'm gonna be. If I'm gonna be in Chicago, then I'll go to ASCO.
Hmm.
Right, that's the one in Chicago that's happening in Chicago.
Yeah, Chicago.
And that's kind of that's as far as I've planned. But I think it ultimately comes down to if I can get an abstract and I have some funding to go, then I'll go.
Hmm.
I think most residency programs provide some sort of compensation for travel, especially if I can get a pathology perspective into the abstract. So
Yeah,
and I won't know where I'm going to be until March 15th.
March 15th. Yes, that's the match day. So you apply for pathology program?
Yep,
Okay. You apply for Northwestern too?
I did.
Okay. So we'll see.
Yeah.
Where's your #1 city you want to land?
I'd like to go back to California just because my family's there.
Oh, okay.
And
Bay
and
Area?
yeah, Bay Area.
Okay.
So either UCSF or Stanford. The
Okay.
nice thing is both of those programs have a really robust digital pathology department, which is really nice.
Yes. There's a lot of
Versus
sequencing companies there, so
yeah,
it will be nice.
versus Northwestern, which doesn't do as much sequencing. Oh, sorry, not sequencing, doesn't do as much digital pathology, but that's okay. Kind of broadly though, I think if I don't end up at Northwestern, end up somewhere else, I'd like to keep working with you. It's just it all depends on availability.
Right. But ASCO would be around the time you start.
Right.
Wait, are you
It's
still
like
student or what? That's kind of first week of June. So
right, first week of June. So I'll be available or should be available.
when's your graduation?
In the middle of May, May 13th.
Oh, middle of May. Oh, so you're not student anymore. Oh, okay.
But I'm gonna see what the alumni options are. I'd like to still have access to all the computing resources and all of that. And
Of course.
I don't know how that works for alumni, or if I'm, affiliated with your lab, maybe I'll have access to it. I don't know.
Yeah. Huh. Yeah. Let's figure out that. Yeah, definitely we can keep working together. And about the H&E slide and genomic heterogeneity, did you have any updated idea? I know it's a daunting task.
No, I did kind of want to try to work within either likely within Tempus's workspace, just because it's something that I think they'll support, and the computational resources are really accessible, and so there's some things I wanted to try. But that all depends on them getting back to us. At the same time, I've got a little bit more time in March, and a little bit of time in April, and so I can start doing some of this on the computing cluster in Northwestern. I've been able to I've gotten to the point where I can take a slide, I can chop it up into pieces, I can highlight the different types of cells that are present in each patch. I can count them. The kind of the next step from here is asking, okay, well, we have to ask a question, or do some kind of higher level study where we say to ourselves, folks with these qualities have these many cells in these areas. Like, we have to ask a question, and then I can start playing with it and coming up with some kind of answer, for quantification.
Yes.
But it's like high level cell type, so it's like lymphocytes, stroma, neoplastic cells, normal
Right.
cells, or
Yes.
like miscellaneous.
So keep thinking. We have the Lunit, we
Right.
work with Lunit, so they are able to calculate, well, draw for us where the stroma is, where the lymphocyte is, where the tumor is.
Yeah.
So we can get that digitalized data where, we and based on that, and then immunotherapy treatment outcome, and with the CT scan, and a lot of them will have our in-house NGS data. So we in PD-L1, we have all that 170-ish. Liam, how many radiomics total did we submit?
160.
160. So that's a valuable data, and
Yeah.
Stas, you have access to it, but we just have to ask the right question. Like, because Lunit only looked at TIL. Was it TIL high or low? That's it. But you want to ask beyond that, like, does the heterogeneity or the structure of the tumor, stroma, and TIL,
Right.
how you assess that heterogeneity, convert that into something meaningful clinically, right? So
Right. Yeah, I mean, I imagine you would have to. Well, we can try. I know there's also algorithms out there that can draw. So most of these slides also have adjacent normal.
mhm.
And so what we could do is see if we can get something to pull out the location on the slide where the tumor is, and then evaluate that tumor section based on where in that section there are immune cells strong, like come up with a way to say, okay, well, it's peripherally rich but internally poor, or it's internally rich and peripherally poor, or some kind of stromal lymphocytic interaction, maybe. I don't know.
Keep reading. It's a
Yeah.
hard area to explore. But I think it's a valid question. We just don't know what the best way to analyze it is.
Yeah.
So the question is, histological heterogeneity, can that be reflected in genomic heterogeneity? And in understanding the association between the two and its interplay, and or combining both metrics, can we improve upon our ability to predict outcome,
Right.
clinically? So that's what matters in the end. Can we use the marker to personalize treatment? So that's where we want to end up in. Okay.
Yeah. I'll keep reading and see if there's, because it all comes down to how we define heterogeneity. Do we define heterogeneity by how much neoplastic cells differ one from the other? Do we define it as the distribution of immune cells through and stroma throughout the tumor? Do we define it? That's really the question.
Or one thing that just came up to my mind is, a lot of times as cancer grows fast, there's a central necrosis.
Right.
And if you do PET, there's like a dark hole inside. Maybe that's indicative of aggressiveness, but also indicative of, HIF, hypoxic,
Inducible factors.
and then all these things, and maybe because the whole slide analysis is difficult for human eyes, but you kind of look at it and let the AI will let us know the necrosis proportion, and
So quantifying necrosis.
Because the HIF also plays a role in modifying the immunotherapy response as well. So maybe that's one way for us to use AI, like necrosis index or something. And then that's another way of assessing heterogeneity. What pockets of necrosis are present.
Right.
And the pattern of of that versus tumor, live tumor, things like.
I mean, it would be interesting to be able to plot, like for example, with mutant allele tumor heterogeneity math, where they have a distribution of variant allele frequencies, and then they calculate how wide that distribution is. I wonder if you can come up with something similar for necrosis, or even for any other metric, and the x-axis is going to be the x-axis can be the dimensions of the slide, and where across the slide you're seeing the presence or absence of certain features, for example, necrosis. And then do that in a 3D way across the slide. And that's how, I don't know. I'm brainstorming here.
Sure. Well, we should keep brainstorming because it's
Yeah.
it's a hard topic. Okay, so let's finish your paper first and let's pursue the Tempest company data analysis. And if you want to add Tempest H&E slide for this project, I think we can
Yeah.
ask them for that image. And we can analyze the image with the you know, digital AI program as well. Okay. So yeah, I mean, if we were asking this question, someone probably already did and made a shareware. So look for that first.
Yeah. Yeah, it's surprising how much freely accessible software there is for machine learning, digital image
Yes.
analysis.
And
It
our
may
faculty, you know, Lee Cooper is
Lee Cooper, right?
yeah, you can meet with him as a future pathology resident and
Right.
kind of pick his brain. He's
I've
already
just been
helping
told
us.
I've been
Yeah.
told he's fairly busy, so I wanna have something interesting to propose to him before I meet.
Right. And oftentimes he works from home. So
Right.
okay. Okay, anything else, Das?
Nope, that's it.
Okay. Anybody else before we wrap up? No? Okay, so we'll call it a day. I'll see you guys. Bye.
Have a good week, everybody.